I wanted to orientate you towards the perireceptor environment. So this is that space in between the epicuticle and then the internal part. And the perireceptor environment is this biochemical physical space, right? And you can see there's a lot of complexity here, but I'm going to break it down for you. And it's a biochemical physical space that surrounds the sensory dendrite, which is the outside, with the internal chemosensory sensilla, which is this one here, right? And so it's got a crucial role because it shapes how chemical signals get captured, how it transports, how it transforms, and ultimately it's the insect nervous system perception that we're talking about here. So we can think of these little circles on currently on the outside of the animal as ligands. Now that's just of course a very broad chemistry term, but in the context of insects, these might be odorants, tastants, and you can see here that they find their way physically through pores that line the epicuticle on the outside, right? And there's a little bit more complexity in structure here, but we're not going to go into too much detail, except that these ligands, and there's a huge diversity of them, of course, enter the pores and then enter this perireceptor environment. Okay. Now you'll notice here there are a lot of these structures shaped like that in this schematic over here. And we refer to these as OBPs. Now what that stands for is odor binding proteins. They are small, they are soluble, and they are secreted into this lymph, right? So the lymph is this idea of like a lymph node in your own body, but the idea of a biochemical space. So they're secreted into this sensillar lymph, so to speak, right? And there are these auxiliary cells that secrete these OBPs. And the function is that they will bind, like the name suggests, odor binding proteins. They can solubilize. Now, if your ligand here is hydrophobic, they will solubilize it, they bind it, and then they change it to a hydrophilic complex. They can also protect these ligands from getting degraded non-specifically. And of course, they can also transport the ligands to specific receptor sites, right? So OBPs, because there's also a subsequent diversity of them, they can contribute to some level of selectivity, right? Different OBPs, they can bind to different classes of ligand. They influence, in doing so, they influence which odorants ultimately reach the dendrite receptors, or what we call the sensory dendrite. So these dendrite receptors, you can see here, they are bound to the dendrite itself, right? They are embedded in the membrane of the dendrite. They are sometimes called odorant receptors, ORs, but that's generic. Sometimes you have versions of these that are gustatory receptors, so GRs, but in this case, we're talking about smell in this particular example. So these would be odorant receptors. So this complex that you see here, this OBP ligand complex, is going to be transported to the dendrite receptors. Then there will, of course, be a response, down that cascades down via the dendrite. But in some cases, there is this that happens, which is they enlist a degrading enzyme, and the degrading enzyme will actually then break down the ligand into other component parts, right? So they maintain some kind of sensitivity and temporal resolution because if I smell something, right, do you notice what you perceive as you are now desensitized to the smell? You don't smell it anymore. After a while, get used to this smell. That's in a weird way because your body has desensitized to it. Now, in the case of an insect, there are some instances where if I smell something, I need to very, very quickly reset my stimulus so I'm receptive to that smell again. You think about the context of reproduction. You think about the context of feeding. Now, if you are stimulated once and then you get desensitized to it, then there are opportunities for feeding or reproduction that you miss if you don't quickly reset your system. So these enzymes do just that, right? So we would describe this more I guess, more formally as maintaining sensitivity, right? Maintaining some temporal resolution, meaning to say that I am stimulated at this time, but then I want to know when I'm next stimulated as well. So as an insect, I need to find a mechanism to rapidly terminate these signals, right? So odor degrading enzymes do that for me. They are also in the sensular lymph, right? And they are there breaking down the ligands after the receptor activation has occurred here. So it prevents the insect from getting stimulated in a prolonged way. So no prolonged stimulation. You've got rapid signal resetting, and then it, as an insect, enhances my ability to detect odor concentrations more sensitively in my environment. So if we integrate all of this functionally together, you've got your ligands, you've got your OBPs, you've got your degrading enzymes, you've got your dendritic receptors. They are all working concurrently, and in integration, they regulate receptors, they enhance sensitivity, specificity, and you've got really, really precise control of chemo sensory signaling, right? So this is quite beautiful. We know a lot about this space. What we don't know a lot about is this space here, actually, and that is how specific particles actually physically move through these pores. But more to come in the future, I'm sure.